Good afternoon, everyone, and welcome to Grand Rounds. My name's Tom Fardman. I'm the chair and organizer of Grand Rounds, and these slides keep moving on by themselves. I apologize. They have a life of their own. Welcome to the people in the auditorium. For those of you on Zoom, it's an absolutely packed out auditorium today, standing room only at the back. Thank you very much for coming in person and for joining online. So I said in my email that I sent this week, Grand Rounds, we try to be as varied as we can be. We try to cater for clinical aspects of things, scientific aspects of things, some more human interest ideas. We have input from our global citizenship teams and sustainability, and even occasionally we have some medical students who wander in and then realize they're in the wrong room and wander out again. It's always an honor and a privilege to have visiting speakers, and I'm grateful to Esther Samler, who has put us in touch with Ai-Huey Tan, who's come all the way across from Malaysia and has managed to fit Grand Rounds into her busy schedule. I was talking to Esther just before this. I'm sure this is what we're going to go into here, but Dundee is a hotbed of research in Parkinson's and movement disorders, and we've had talks before from Miratul and from Esther about this and some other visiting speakers. But what's interesting is that a lot of the research naturally is done on our local population here of Caucasians with movement disorders, and what we don't know necessarily is if that evidence and research translates into the non-Caucasian population, and I think that's very much where the collaboration with Ai-Huey comes in. Now, you're going to talk about something different, I think. Well, it's going to talk about Parkinson's disease, but you're going to talk about the gut microbiome. I'm a chest physician, and 10 years ago, we thought that the lungs were completely sterile. And if you think about that, that's an incredible shift from what we know now. The gut was dirty and the lungs were clean. That's what we always thought. Don't put the bronchoscope down the wrong hole. It's dirty down there. And now we know, of course, that the lung microbiome is really, really important in terms of lung health, but not just lung health, but overall health. Visiting all the way from Malaysia, thanks very much for coming. Spontaneous round of applause in the auditorium. You can click with this. You can move your pointer with that.
And
we're all good.
This mic?
You get that microphone,
right.
And the camera's pointing right at you.
Okay. Thank you very much, Professor Tom. Hello, everyone. I'm Ai-Huey Tan from Kuala Lumpur, Malaysia. And I just want to start by thanking all of you for coming to this talk. And I really especially want to thank Dr. Esther Samler for hosting me and my team in this one-week research visit in Dundee. It has been absolutely amazing to see, the work that has been done by Dr. Esther's team and Prof. Dario's team in their lab. And I just want to say also that we really appreciate how kind the team has been to us, to the small team from Malaysia, who's also starting out to do more and more research in Parkinson's disease. In our center in Malaysia, we have maybe two main, okay, now I can see why we get distracted as the slides keep moving on its own, that we do have 2 or 3 main domains of Parkinson's research in Malaysia. One will be the genetics of Parkinson's disease in our population. The second one will be on the role of the gut in Parkinson's disease. And the third one will be, discovery of biomarkers related to our population as well. And we've been very fortunate to collaborate with Esther and also Professor Darius and team on the LAC2 Parkinson's Asian variants in our population. But today I will be sharing with you on another domain of our work, which will be on gut microbiome, Parkinson's disease. And in this talk, I aim to summarize not just the work that we have done in our center, but rather, the work that has been done by many, many wonderful scientists and clinicians around the world on this important topic on the gut microbiome in Parkinson's disease. I have nothing to declare for this lecture, but here I think that because we have a wide spectrum of audience, so I think that it would be very good to just start with an introduction to the gut microbiome brain axis, and I'll give you an overview of the clinical gut microbiome studies in PD, and then we'll discuss the role of the gut microbiome as potential biomarkers and therapeutic targets in PD. So let's start. So as you may know, the GI tract harbors several trillion microbial cells, most of which are concentrated in the large intestine. If you compare this number to the number of host DNA cells that we have in human, you actually have 10 times more microbial cells in our body compared to human DNA cells. And the gut microbiome is a very interesting topic. The colonization begins at birth, as you can see in this picture, and it actually evolves through different stages of life. The microbiome changes as we change our diet from breastfeeding to solid nutrition, as we go through hormonal changes during puberty, and as our GI tract also mature and proliferate. And when we age, there may be some low-grade chronic inflammation happening in the gut that will then lead to further changes in the microbiome. And throughout our lifespan, the gut microbiome play critical roles in our anatomical, physiological, and immunological development. And sorry, somehow the slides just keep moving on its own, but never mind. But if you look at this picture, you'll be wondering how to study something that keeps evolving throughout a person's life. But I would like to share that the adult microbiota here, as I highlighted in this green box, is relatively more stable, because at this stage, we then develop a core community of permanent colonizers in our gut with very high functional capacity. And as we age, the bacterial diversity in the gut reduces, with consequent reduction in the functional capacity. So I think that when we want to look at the gut microbiome in neurodegenerative disease like Parkinson's disease, where it primarily affects older people, then we need to understand the changes of gut microbiome in the elderly first, as summarized in this picture. And I just want to draw you to two very fascinating studies that I found in this field. Firstly, there were studies that have looked at the gut microbiome of people who live more than 100 years old, and they found that these people had more diverse gut microbiome compared to other elderly people. And this gives rise to this concept that maybe if you have a more diverse gut microbiome, it's supposed to be very good for your health, because then you have different bacteria doing different function for your body. And then they also found differences between elderly people who are community dwelling, which means that they have still a fairly healthy and independent lifestyle, compared to those who are living in long-term care facilities where they are more frail elderly, and their microbiome is also correlated with increased markers of systemic inflammation. And this sort of alteration in the gut microbiome composition and function that is associated with an unhealthy state is then termed as gut dysbiosis. But really what constitutes a normal microbiome for a human being remains undefined. So there's a lot to do in this field. And I think that the other interesting fact about the gut is that it's actually the largest body surface area that's continually exposed to antigens and metabolites from the external environment. There's a recent study that has actually measured the surface area of our entire gut mucosal epithelium. Guess how big it is? It's the size of half a badminton court. So that's quite big. You have half a badminton court stuffed inside your whole gut. And this half badminton court actually also contains the largest microbial density you have in your body. So that means that the gut is a very vulnerable port of entry for pathogen. And because the gut microbiome play critical roles in gut barrier defense system, as you can see in this red box here, when you have an alteration in the gut microbiome composition, then you might have an alteration in your gut barrier defense. And therefore, you may have increased gut permeability and inflammation happening at the mucosal level. And this leads to the condition what we term as the leaky gut, which then may enhance translocation of pathogen and toxin from the external environment through the mucosal epithelium and through maybe your blood circulation, and then go into your systemic processes in your body. And the other very interesting finding that I would like to highlight here is that people have also found that the terminal endings of the vagus nerve in the gut mucosa are mere micrometers away from the gut lumen. So you can see that the end terminals of that vagus nerve is just, a few micrometers away from the external environment. And then this mucosal barrier is like the defense system between the external environment where you may have pathogens and toxins to your enteric nervous system. So this raises a possibility that an exposure of an toxin or infectious agent that can then leak through the gut barrier and then spread to the brain through the vagus nerve. And therefore, you see, in the last decade, there has been a lot of work on the microbiome gut-brain axis, not only in Parkinson's disease, but also in other neurodegenerative diseases like Parkinson's, like Alzheimer's, ALS, epilepsy, as well. And we believe that there is a bidirectional interplay between the brain and the gut through neuronal signaling, immune pathways, metabolic pathways, as well as endocrine pathways. Now, in the clinics, being a neurologist, I think we also want to see what's happening to our patients with Parkinson's disease in the clinic as well. And we found that GI symptoms are very common among our patients, and it can occur along the whole GI tract. This nice diagram here by Ron Pfeiffer summarize all the different GI problems that a patient with Parkinson's disease can experience, and up to 80% of our patients experience constipation throughout the disease course. And what's really interesting is that constipation can also occur as a prodromal symptom predating the mortal diagnosis of PD by several years, and constipation has been associated with a 2 to 4-fold increase in the risk of future Parkinson's disease. And the other very important discovery in this field of gut microbiome brain axis in Parkinson's disease lies in the discovery of Lewy body, the neuropathological hallmark of Parkinson's disease, in the enteric nervous system of patients with PD. As I have written here, autopsy studies have demonstrated alpha-synuclein deposition, which is a neuropathological hallmark for Parkinson's disease, in the enteric nervous system in almost all individuals with PD. And in clinical gut biopsy studies with collaborations with the gastroenterologists, then such scientists and clinicians have also found that alpha-synuclein deposition can be detected in the esophagus, stomach, small intestine, and colon up to 20 years before PD diagnosis. And this is very interesting, something that we should really look furthermore. And a recent multimodal imaging studies found that patients with prodromal PD have marked peripheral autonomic nervous system involvement despite a relative sparing of the nigrostriatal dopamine system in the brain. So let's maybe zoom in a little bit more on the specific role of gut microbes in PD pathogenesis, there has been an emerging literature from several preclinical and clinical studies that have demonstrated the role of gut microbes, as you can see in these red boxes here, or their microbial products, mainly amyloid proteins like curly or lipopolysaccharides. They can promote gut inflammation, in part via activations of toll-like receptors at the gut mucosal epithelium, and this can lead to neuroinflammation, alpha-synuclein brain pathology, and or motor dysfunction. Many of these are done in mice model comparing wild-type mice versus germ-free mice with alpha-synuclein overexpressing model. So here and the gut microbes is postulated to not only play a role in PD pathophysiology, but they can also play a role in PD drug metabolism. And one of the main drugs that we use to treat our patients with Parkinson's disease will be levodopa. And earlier studies have suggested that Helicobacter pylori infection, as you know, this is an infection in the stomach, may reduce levodopa absorption, but more recent studies show conflicting results. And a very recent study, or not so recent now, found that a bacterial enzyme in the small bowel called tyrosine decarboxylase can convert levodopa to dopamine in the small bowel. And this has been a very important finding in the field in the last few years, because what happens here is once levodopa is being converted to dopamine in the gut, dopamine is postulated to reduce GI motility, and this can lead to a condition what we call a small intestinal bacterial overgrowth, and possibly overgrowth of the enzyme TDC, enzyme producing bacteria, causing a vicious cycle in this process. And this will also then reduce further the levodopa absorption in these patients. And in our center, as well as research teams in other different centers, we have also found that Helicobacter infection and small intestinal bacterial overgrowth have been associated with worse Parkinson's disease severity, and this might be one rationale for this observation. Now, I just want to maybe go down then to the clinical studies of gut microbiome Parkinson's disease. I want to start with this diagram. As you can see, the distribution of the kind of bacteria we have in our gut is not uniform along the whole GI tract. And the highest density is seen in the colon, because the colon has a very nice pH and a very nice environment for these bacteria to grow. And therefore, it's not surprising that most of the studies on gut microbiome in PD are done using fecal sampling, where we have more than 50 studies in the field right now. And when I started my research career, I have never thought that I will build a career collecting fecal samples from patients. And now this has been somewhat like a goal for me every time we collect a stool sample from our patients. And remember that most of them has constipation, so it's not easy to collect the fecal samples in patients with constipation. And in our center, we have this very nice drop-off pickup service for your fecal samples. So when you've done your motion, you text our research assistant, someone will drive to your house and collect just stool samples, rush back to the lab to freeze store them immediately in -80. Now, this is a very rewarding journey actually, because the fecal samples contain so much bacteria for us to study. It gives us a very non-invasive and cost-effective method to provide a snapshot of the colonic microbiome. And this is just a slide that I have done in 2018. We have only 10 studies around the world at that time. And fast forward to 2022, you have 5 times more the studies that you have seen in 2018. So it looks like everyone in the world loves collecting fecal samples from patients with Parkinson's disease. And I wonder if someone from Dundee would start doing that as well. And if you would like to, please contact me because I will be very happy to share you the collection protocol as well as the clinical data pro forma. Now, I just want you to have a look at the colors of the X I've put in this map. So you see many red X's. These are studies that are done using 16S RNA gene sequencing. This is a relatively inexpensive method. It provides resolution mainly at genus level. But I must say that this is a limitation because if you look at our gut microbiome, really between two different human beings, the variations actually is at species or strain level. So we need to actually go down with a higher resolution sequencing to really understand the composition of gut microbiome. And as you can see in this map, the number of studies using higher resolution shotgun metagenomic sequencing remains quite limited in the field. And this is because it's really very expensive and the bioinformatics can be also very challenging. Now, despite all the different technical variations in the sequencing regions or technologies that are conducted in different centers around the world, almost all studies found a significant difference between the gut microbiome composition of the PD patients versus controls. And our team decided to meta-analyze, these available gut microbiome sequences in the public database. You see here a meta-analysis of 1,700 subjects. And we actually found that if you look at the first plot here on the top right corner, you can't really see a very big difference between the red dots and the green dots. And this is because that the PD status only explain about 0.4% of the variances in the gut microbiome composition. I think this is a very important concept to appreciate. Although there is a significant difference between the composition between these two groups, but the differences is actually quite small. And differences in study, geography, explain a larger proportion of these variances in gut microbiome. This is about 17% in our study. Why is this so? This is because various intrinsic and extrinsic factors influence our gut microbiome, and this include our age, frailty, comorbidities, slow transit time. If you have constipation, your gut microbiome might differ, your BMI, diet, physical activity, and medications. So interestingly, the effect of non-microbiome covariates is surprisingly small, and most factors explain only a single-digit percentage of observed microbiome variation, suggests that actually there are also further important uncharacterized covariate factors that we have not found. And we still need to look at this further. And numerous studies have then now controlled for all these confounding factors. And interestingly, they found that the diagnosis of PD still have a very significant effect, a significant independent effect on gut microbiome composition, even after confounded analysis. Now, I think some of you then will ask me, how about medications? How about the medications taken by patients with Parkinson's disease? So there are some studies that have found the usage of COM inhibitor, and if you are a Parkinson's neurologist, you might have used these medication frequently in patients with motor response complications, and you know that one of the side effects is diarrhea. And they found that this has an influence in the gut microbiome, correlating with our clinical observation. And there are two large case control cohort studies that have found significant difference in the gut microbiome between de novo PD patients, that means patients who have not been treated with Parkinson's medications, versus control. So there is a signal here between the disease patients and versus healthy controls. Now, although that all the studies found significant difference in the overall gut microbiome composition, but they are quite heterogeneous in terms of the specific microbial changes in their cohort. And therefore, it's very important then to meta-analyze the data and put everything together and see if we see any consistent signals. So these are two meta-analyses, one by the group of Romano et al. And one by our own group here in Malaysia. And we found that there are several more consistent signals. And this is characterized by an increase in lactobacillus, bifidobacterium, and also Akkermansia, and the reduction in Lactospiraceae and Roseburia. And interestingly, these findings from the 16S RNA meta-analysis also were quite consistent with findings from a recent meta-analysis of shotgun metagenomics, the higher resolution sequencing platform. So then, the question of the day, have we now found a new diagnostic biomarker for Parkinson's disease since we have consistent microbial signals, right? Now, I think if you look across all the 50 studies regarding gut microbiome in PD, you'll find that increased Akkermansia will be like probably the most consistent microbial signal. And I have summarized here studies that have found this. But if you expand your literature review to include other neurodegenerative disease like multiple system atrophy, progressive supranuclear palsy, Alzheimer's, or amyotrophic lateral sclerosis, you find the same signal as well. And also increase in Akkermansia has also been shown to be increased in individuals with constipation with a prolonged colonic transit time. And this brings us to this notion that many changes found in case control studies may not be disease specific. It may be just a non-specific shared response of health and disease. And therefore, it's very important to now look at the comparison in gut microbiome between PD patients with a positive control. For example, patients with MSA or PSP, which are atypical Parkinsonian disorders. And I'm showing you two studies here that, one didn't really find any difference between PD patients and MSA and PSP. One found that they were able to discriminate between PD versus MSA versus AD, and we really need more literature comparing these groups of neurodegenerative diseases. Now, then
when we look at microbiome changes in PD, is this a case of increase in PD?
Oh, okay. So I think, how do I do this? Okay, so I think back then I recorded one of the lecture for the MDS Congress, and I think that maybe that slide contained a recording from that lecture. I'm so sorry. What I wanted to ask, but I think that woke everyone else up as well. So, so I think the question I wanted to ask is that when you look at these signals, is this a case of increase of good bacteria in your gut and reduce reduction of bad bacteria in your gut, or vice versa when you talk about health and disease? So several texts are found to be consistently increased in PD, are actually widely regarded as potentially beneficial rather than detrimental. So it's very interesting when you look at this fact, because then why would patients with Parkinson's disease have increased beneficial gut bacteria? And I think that then we need to really understand that, sometimes certain bacterial species can have mixed results of beneficial or harmful effects in different diseases. And there remain many whose function in human health are unknown, and bacterial function is likely to be strain specific. So many of the studies that I've shown you are at genus level. We have not really gone down to species and strain resolution. And here is an example why a strain resolution is so important. If you look at E. coli, different E. coli strains can be either detrimental and catastrophic to your health, or can be rather beneficial as well to your health. So I think that we should really go down to the strain level by doing more shotgun metagenomic study. And this is one of the largest study we have in our field now, 724 subjects, and they found 84 species to be significantly altered in PD. 10% were identified at strain level, and using these, they were able to predict certain upregulation of immunogenic pathways and downregulation of neuroprotective pathways. But there are also caveats to this. I want to say that a significant proportion of the genes in the human microbiome cannot yet be functionally assigned. So we actually do not know, what are their transcriptional and translational activities. And this is very nicely demonstrated in this study on, remember I showed you that a recent study found an enzyme can convert levodopa to dopamine in the gut by this TDC enzyme. And in this particular study, they predicted that 24 E-lenta strains, which is the one highlighted in the red box here, can actually convert dopamine to pyramine because they have the gene encoding the DADH enzyme. But in actual functional in vitro studies, they found only 10 strains were able to do this. So this strain level variability was found to be due to the presence of single nucleotide polymorphism that inactivated this enzyme. And these findings suggest that mere detection of bacterial genes may not accurately predict metabolic functions and highlight the importance of probing bacterial function directly. And I also want to highlight this notion that it is very important to note that the change in the gut function is likely not to be due to an alteration of a few species in our gut, but rather it is the net result of a very complex interactions between the microbes and microbes, the host and the microbes, and the host microbes and the environment they live in. So you can see that this is actually a very complex pathways that are happening in our gut, that we might not be able to solve it with a very simple case control study or a simple mice model. We need to also have better models to study these complex interactions. And I want to encourage all of us now to then maybe think about the gut as a community. You have a nice community in your gut, okay? And when this community composition undergoes changes due to perturbations, then the collective function of the microbiome also changes. This picture summarizes the function of the gut microbiome here again. And scientists have also found that despite the high inter-individual variation in microbiome composition, for example, for you in Dundee and for me in Malaysia, if you compare my gut microbiome composition and your gut microbiome composition, we might be, completely different because we probably eat, very different diet. But then if we measure the functional capacity of my gut microbiome and your gut microbiome, we might be fairly similar if we have similar health status. So here they have shown that the functional capacity between different individuals of the gut, the functional capacity of the gut microbiota can be relatively consistent, and therefore gut functional markers such as gut metabolites may be more promising as seen in other fields. And I want to highlight this as an example in the field of cardiovascular medicine. The gut microbiome metabolite called trimethylamine N-oxide is emerging as a very good biomarker for disease in cardiovascular diseases. So I think that we should really move from, evaluating who is there in the gut and predicting what they can do to really looking at what are they doing in the gut. And this really requires a multi-omics approach. And if you want to study the function of the gut microbiome, then fecal metabolomics may be one of the best way to look at this functional readout. You can actually evaluate these through different platforms like GCMS, NMR, and LCMS. They all have advantages and disadvantages because they pick up different form of metabolites. And I think that most people then will recommend that you do at least 2 platforms so that you can pick up as many different metabolites as possible. And we have done this as well in our center, and as well as other scientists in this world as well. We actually also found differences in the gut metabolomic composition between PD patients versus control. Many of these differences were in the lipids, amino acids, vitamins, and other organic compounds. But I think the most consistent metabolic feature will be the reduction in fecal short-chain fatty acids, such as butyrate, acetate, and propionate in PD patients versus controls. Here is just to show you an example of the fecal metabolomic study in our cohort. And I just want to then point out that we have also done a similar fecal metabolomic study in patients with multiple system atrophy, which is a Parkinson plus syndrome versus control. And you can see again, there are overlap in fecal metabolic signatures. So again, I think that this may be a shared response in coping with disease and health. So we still need further studies to uncover these potential fecal metabolic studies. Lastly, before I move on to another section of this talk, I want to highlight that Parkinson's disease, as we know, is a very heterogeneous disorder. And there are differences in the type of phenotypes we see at presentation, and during the disease course, some patients do very well with very little medication for many years. Some patients develop motor response complications very early. They have to rely on advanced therapies. They progress faster. So this suggests that there may be differences in individuals with PD in the starting point, in the patterns, speed, and extent or spread of PD pathology. At the moment, with the recent multimodal imaging studies, a body first versus a brain first Parkinson's disease hypothesis has been proposed. And I think that if we one day find a very good consistent gut biomarkers, it can also be only relevant to a subgroup of patients who may have a body first phenotype. Now, can the gut microbiome now be new prognostic biomarkers for Parkinson's disease? Can we use the microbiome or the metabolite to predict who will progress faster or who will progress slower? So here is a summary of studies that have been trying to find association between the different motor features and non-motor features with various differences in microbiome signature. But these are all single studies. They have not been replicated. I would not dwell deeper into them, but I just maybe want to highlight one potential biomarker for disease in Parkinson's disease, and this lies in the short-chain fatty acids. So what are short-chain fatty acids really? These acids result from bacterial fermentation of dietary fiber, and they are key players of gut barrier integrity, immunomodulation, and endocrine signaling. And in mouse models of PD and Alzheimer's disease, butyrate, one of the major short-chain fatty acids, has been shown to be neuroprotective. And in Parkinson's disease, various studies have found the association between short-chain fatty acids and risk of PD conversion. So if you have lower fecal short-chain, lower short-chain fatty acids producing bacteria, you may have higher risk of conversion to Parkinson's disease if you have a prodromal phenotype like REM sleep behavior disorder. And in patients with Parkinson's disease, various studies have found the association between short-chain fatty acids with disease progression, with motor progression, as well as with cognitive function. So for example, in our study, we have found that patients with lower cognitive scores also have lower fetal short-chain fatty acids. And I think that the literature on short-chain fatty acids is not just unique or special to Parkinson's disease. If you look at the literature of short-chain fatty acids in other diseases, you might find the same one as well. And here is another study showing lower fetal butyrate levels correlate with worse depressive symptoms. Another potential biomarker will be maybe fetal or serum trimethylamine N oxide. But here in the current state of literature, this remain, study in a smaller population, and the results are conflicting. But I think that it's still very worthwhile to look further into TMAO in PD as well. Now, there is also another potential simple biomarker that we can look at, which is fecal calprotectin. This is a surrogate marker of intestinal inflammation. And if there's any gastroenterologists who are logging in today, you will be very familiar with this biomarker, because this is now FDA approved biomarker for monitoring activities in patients with inflammatory bowel disease. And in several studies now, we have found fecal calprotectin to be increased in PD patients versus control. And in our own cohort, we found similar pattern, but we also found fecal calprotectin to be even higher in patients with multiple system atrophy and other Parkinson plus syndrome. So you can always coming back to this notion that there is an overlap in these signatures, and these may not be very disease-specific signatures. I think it's really important to understand this. And then last but not least, fecal TDC gene abundance. Remember, I spoke about the enzyme that can convert, that is responsible for levodopa metabolism in the gut. And this can also be a potential marker for levodopa response, as shown in several studies here. Now, I want to quickly go through, this very important question. Can the gut microbiome be new therapeutic targets for PD? Because I think this is probably the most important question for our patients in the clinic. And we can modulate the gut microbiome through several different ways. One through diet or prebiotics, or you want to modulate them through supplementation of probiotics, fecal microbiota, or antibiotics, or would you like to supplement them with the beneficial gut metabolites using small molecule biologics? Now, with regards to modulating the gut microbiome to improve levodopa absorption, I think maybe we are getting closer here to the answer. So while earlier small trials suggest Helicobacter pylori eradication may improve levodopa absorption, in our center we did a RCT on this in a double-blind placebo-controlled manner. We found HB eradication did not improve UPDRS motor scores, as well as any of the secondary outcomes in this trial. However, this trial is also limited in the sense that we only had a 1-year follow-up for this trial. So probably a longer follow-up to see if there is any differential signals in the disease progression will be very important to understand if there is an effect of HP eradication in Parkinson's disease. But here is where the answer is getting closer, because in the same experiment that they did that found the enzyme responsible for levodopa metabolism in the small bowel, they also found that a small molecule inhibitor called AFMP can then inhibit that TDC enzyme. And I think that we should see, I look forward to see further work on this, because this has very, very direct translational benefits, that if you supplement your patients with levodopa and this enzyme inhibitor, then the levodopa absorption may be improved, and that may really help the patients. The other, of course, valid question is, if we eradicate small intestinal bacterial overgrowth and try to reduce the number of bacteria that produce this enzyme, will this work? But but I think the literature regarding SIBO eradication in PD remains very limited and probably worthwhile to be explored further. Now, the role of diet in PD risk and progression have demonstrated mixed results with some discrepancy between prospective versus case control studies. We have summarized those literature in a table format in our review article. But I think the most exciting or most consistent results from the diet studies in PD will be the association between caffeine intake and inverse PD risk. This has been highly reproducible across several well-designed studies. And if any one of you who are logging in today, who are coffee lovers, this is great news for you. And I just want to say that in these studies, I think you probably need 2 cups of coffee to get the effect, but even better if you drink 4 cups of coffee a day. So, and also there are some investigators who have also looked at a Mediterranean diet, and this has also been associated with lower probability of developing prodromal symptoms of PD. However, adequately powered RCTs on diet as treatment strategy in PD remain lacking. Now we move on to prebiotics and probiotics, there's a huge potential for probiotics because the mechanisms are so diverse, as you can see here in this little diagram here, and there have been many studies that look at probiotics as a treatment for constipation in PD, including the one done in our center. And I'm very pleased to share with you that we now have class 1 evidence for probiotics as a treatment for constipation in PD. Now, I think that many of our patients do come back and keep reporting that, it is, they have been very pleased with the probiotic supplementation, really a very simple, non-invasive way to treat those very bothersome constipation that they have been experiencing. And in my clinic, sometimes I will also advise them to buy the type of milk powder now that is formulated with prebiotics or probiotics, so they actually, can gain weight, pump up their nutrition, and pass motion very well at home at the same time, keeps them very happy. So but now, I mean, we are all very excited about probiotics, but I think that we also need to understand that the usage of probiotics far outpace the scientific understanding for their effects. And we still need more mechanistic studies to understand why they work, how they work. We need higher quality, adequately powered RCTs with longer follow-up data, and we need to look at safety data. This is just a very recent paper published this year on prebiotic fiber, and this is an open label study in a very small cohort. They did show some improvement in constipation, but I was just, quite interested in the diagram on the right here, where they were able to show that prebiotics intervention actually, have effects on plasma zonulin, which is a marker for intestinal permeability, on calprotectin, which is the marker for intestinal inflammation, and also neuroferritin light chain, so which is a marker for neurodegeneration in various diseases. So it's really quite interesting. And last but not least, fecal microbiota transplantation. If you're wondering how this works, you can either have it done through an endoscopy method, or you can have it packaged in an oral capsule to be ingested. Whatever your preference is, doesn't matter. But at the moment, some small studies found benefits in treating constipation in PD, but I think we still need larger clinical trials, and these are all the ongoing trials on fecal microbiota transplantation in PD. So where do we go from here? I think this picture that I have shown you in the beginning of the lecture today actually look more like this. It's a really complex field, and we need to appreciate the complexity, because if we have a simplistic understanding of the role of gut microbiome in health and disease, I don't think that we will be able to build that kind of models that are necessary to understand that role, that function of the gut microbiome PD. There remain several gaps in our literature. We have summarized some of the future directions for PD gut microbiome research in this review article. If you are happy, do have a look at this. I think importantly, I think we need more multinational collaboration given the diversity of gut microbiome we have among different populations around the world, as well as a very strong interdisciplinary clinician-scientist collaboration to make this dream come true, that maybe gut microbiome can be a new frontier for diagnosis and treatment in Parkinson's disease. It will be a dream come true for me and my patients and your patients around the world. And I want to take this opportunity to thank, all the incredible mentors, collaborators, and research team that I have in this journey of exploring the gut microbiome. I was just sharing with Dr. Esther Samler and Prof. Dario's team a few days ago that it wasn't easy for us to embark on this journey. We have a lot of challenges, but I'm glad that, we have learned immensely from this journey, and I'm so glad to have the opportunity to share a little bit of our journey with you today. Thank you.
Mikey, I'm a bit blown away. I don't know what the rest of the audience feels. That's what a comprehensive tour de force presentation from start to finish. I think I'm just blown away. I was just reflecting there about how old I feel, actually. One of my kind colleagues just before the meeting asked me when I'm going to retire, which is perhaps a little bit premature. But when I was at medical school, we hadn't finished the genome project. The human genome project hasn't been finished. And you're talking now about shotgun metagenetics of, what a different world we live in today. Anyway, maybe I should retire, Tom. Maybe I should. Absolutely phenomenal work. And what was striking, actually, just looking at your references on the side of your slides, is how much of this work happened between 2020 and 2022, when the world was locked down due to COVID, and yet you and your colleagues were just smashing through all this stuff, well, smashing through poo by the sound of things, but smashing through it all, making this incredible work. And those papers weren't published in the octosugal journal of the left ear. They were Science and Nature and, proper science, proper journal. So kudos to you. Absolutely, chapeau. I have to get to clinic in 7 minutes. Does anyone have any burning questions? To be honest, I'm quite happy to just wrap things up there. I think that was an incredible talk. And if anyone in the audience physically wants to speak, I'm sure you'll spend some time. There was no questions on the chat, but there is one question. Will you answer one question from the chat? Is that all right? It says, that was a really great talk. Could you perhaps comment if gut microbiome studies are able to make a distinction between Parkinson's diseases and dementia with Lewy bodies? Similarly, given the occurrence of asymptomatic pathology in the olfactory bulb, what is known about nasal microbiome and are changes here likely to be indicative of olfactory bulb synuclein pathology? Big breath. Thank you to who's given us that question. We should thank them for, and that's David Cross. Thank you. Okay. On you go.
Thank you very much for this very interesting and important question. Firstly, about the first question on the gut microbiome studies between PD and dementia with Lewy body. Actually, I have not seen a good case control studies on this, and I think maybe this also maybe one of the challenges will be to recruit patients with dementia with Lewy bodies. And I think that if anyone has a good cohort of patients with dementia with Lewy body, I think it's really worthwhile to look at the differences in gut microbiome between the two. And I think it's a very important question because they seem to carry similar pathology, but then, they have very varied phenotype, and perhaps, there must be something to do with, environmental factors in the phenotype differentiation between the two diseases. Now, on the second questions about nasal microbiome, there have been some studies on nasal microbiome that have shown conflicting results. I would not be able to summarize them here, but I will be very happy if you drop me an email. I can email you the studies on nasal microbiome, Parkinson's disease versus control. Whether they are indicative of olfactory bulb synuclein pathology, I think that in these studies, they only look at the nasal microbiome without further correlation with, let's say, any biopsy samples. But I think this is also another very interesting hypothesis to be proven. Thank you.
Thanks very much. So I said at the beginning, we try to be diverse and across different specialties and different aspects of medicine. And this talk, we've gone through neurology, GI, we've talked about genetics, we've talked about microbiology, we've talked about the nose just now. So we've been across a whole gamut of things. So all that remains to say is to thank you. She's still busy here beavering away typing answers to questions, so I can't stop her. She's a machine.
No, I just wanted to put my email there so that I can email. Yeah.
I think it's this keyboard, but right. there you go. So thank you very much for your talk. Thanks for coming. Thanks in the audience. Next week, a legend returns. John Dick, who spent a career here being a general physician in the truest sense in NHS Tayside. He gave his retirement talk at Grand Rounds in this just right here 6 years ago, but he couldn't be stopped either. So he spent the last 6 years in Australia, and he's coming back, and I've talked him into giving his post-retirement talk to talk about the differences in medicine between Australia and here. So please do come to that. We're going to do it in person back here in the Gannicke Trust Lecture Theatre. So please, if you want to see John, I'm sure lots of people will want to see him and just say hi. But we'll be back in here next week at 1 o'clock. And as usual, it'll be on Zoom if you can't make it. Thank you so much. Some spontaneous round of applause. Take care.